Happy to host you again. We were very happy to read the messages in the WhatsApp. It's beginning to be interesting there. And yeah, we invite you to be there with news, with photos. I would like to see some photographs from your projects, from your farmers, from your fishes. Okay, let's try to share your experiences from there. Okay, we have today an interesting day, another interesting day. First of all, I would like to introduce you to our lecturer and expert, Mr. Achikam Gissis. We call him Achik, and we are so happy to have you here with us, Achik.
Thank you so much. I'm so happy as well.
Yes, I was looking that we met for the first time, I think in 1997, when I did the first fish farming course here in Shefaim, and you were with us. So, wow, it's a lot of time already.
Yeah, I was so handsome that days. Yeah, things are changing.
We were all so young. Okay. But you are very lucky because in the meantime, Achik really did, I think, hundreds of projects, and he has been visiting lots of countries. So he has much more experience now than he had at that time. So you are lucky to have him now for 2 meetings. And his specialty is for sure fish reproduction and fingerling production, but he will tell you much more about that. Okay, I only want to remind you that after Achik's lecture, we will hear about Zambia today, okay? And then we will also split into groups like last time for a little discussion for each country. So without more words, don't forget to write questions in the chat or to raise hands. We will do some stops in the lecture for questions. Okay. So, okay. Please, the mic is yours.
Evelyn, thank you so much. Good afternoon to all. Before I start, I have to apologize. It's my first lecture using Zoom. Always try to do it, eye contact, touch everything. So I hope it won't be too hard and not too many bugs. So let's start. Yeah, I have to share screen probably, Evelyn.
Yes.
Okay, I have put here a short file which will describe what are my plans. Okay, so it's written here, good afternoon and so on. Today, we shall have 2 lectures.
Achik, only open it, please, for a presentation. Make it big.
Who, me?
Yeah, make it big. Make it
a little bit bigger.
It's not presentation. It's just Word document.
Ah, sorry.
Okay. And just to show what I'm planning. So first lecture will be about the general background of reproduction, namely how hormones control and regulate all this process of reproduction in fish. Okay. We should talk about systems and protocol, how to spawn fish. It's on a second lecture. And I will have to say some word about the nursing stage, which I think is so important in aquaculture and not so well practiced in many African countries which I have visited. After that, we shall have, of course, questions and short discussion. We shall have a discussion between the two lectures as well, okay? Some 5 to 10 minutes about what I have said. Just to introduce myself, Evelyn did it very nicely, but currently I'm a fish farmer. I own a fish farm here in Israel. I'm culturing edible fish, ornamental fish, and fish for ecological purposes, to stock in reservoirs, bodies of water, just to control all the pollutants, the organic pollutant like mosquitoes, algae, zooplankton, phytoplankton, and so on and so on. Okay, we stock in Israel fish, not using chemicals. So I'm producing these fish, and of course, ornamental is for export, Europe mainly, overseas. And edible fish, I'm culturing a lot of common carp, gray mullet, some other species. Lately, I quit tilapia because it's not so economic anymore here. So with not too many words, we shall go to our first lecture, and that will be the hormonal control of reproduction. Is it okay? Let's make it a little bigger, maybe. More than that, I cannot. Hormonal control of reproduction. Some definitions. What is hormone? Hormone is a chemical substance.
Wait. Achik.
Yep.
You have to share again.
You have to stop share.
You have
share again.
Whatever you say, Evelyn.
Yeah. You have to open your PowerPoint. That's it.
Is it shared already?
Yeah.
I told you I will have some, technical problems.
It's fine.
We shall overcome. As John Lennon said in his beautiful song, we shall overcome. Okay, so hormone. Hormone is a chemical which is synthesized somewhere in a secretion organ, and it goes to a target tissue within the body, usually or always through the blood system. Okay, this is hormone. There are all kinds of hormones. Some of them are proteins, some of them are glycoprotein, many types of, but this is the definition of hormones. When we're speaking about neurohormone, and this is very relevant to fish spawning, it's usually smaller so-called hormone or neurohormone, which is secreted from a nerve cell, brain or nerve cell, okay? Again, to a target tissue. It can be direct through the nerve to the target tissue. In a minute, I will emphasize a little more, or through blood system and so on. Another group of chemicals are pheromones. Pheromones are, again, kind of hormones which are secreted from one body into the environment and is detected by another creature, let's say from male to female, and so on and so on. Okay, so that's when I will use these terms. I hope now everybody will get it and know what I'm speaking about. I'm asking clear, and I pretend to hear yes, of course, clear. Okay, because I cannot hear you. Now, very, very important. Any hormone needs a receptor in the target tissue which will exactly fit to the hormone. We are talking about its structure, yeah? So a receptor for one hormone is different from a receptor to another hormone. That means a hormone cannot stimulate any part of the body, any tissue, unless it has its receptor to detect the hormone and start all these events which coming later on. It's very, very important, since in some cases, and I'm giving an example even now, some cases like common carp, yeah, Common carp hormone, in a minute we shall speak which exactly, can stimulate spawning in many other fish which are not common carp, because the receptors are enough, sufficient to detect the hormone. But let's say in tilapia, no receptors which can detect hormones of common carp. So it's something very unique, very fine, and we can play with it while we are going to spawn fish or to stimulate them to spawn. Now let's see how this system is, what is composed of. So first and most not important is the hypothalamus. Truly, hypothalamus is part of the brain. Okay? The hypothalamus is part of the brain, in charge of many activities. I'm concentrating now only about reproduction in fish, okay? And the hypothalamus is controlling and stimulate a very important organ named pituitary. Hypophysis, pituitary, okay? The pituitary is sitting under the brain in the upper side of the mouth. I will not put my finger inside because I cannot talk, but anyway, it's a small organ, very important. Control of so many activities, metabolism of, but also very important in related to fish spawning. Another organ, which all of us we are familiar with, are the gonads, okay? In female gonad called ovary. That's, how can I say, it's this bag, sac, which accumulate all the future eggs. In male fish, or not only fish, it's testis, okay? Where the sperm is secreted, produced, and then later on secreted. So these three organs are the hypothalamus-gonad axis, which we play with when we want to induce fish to spawn. Let's go and see what are the relations. I will, one more thing, yeah. The hypothalamus secrete a very small molecule named, as you can see, GnRH, gonadotropin releasing hormone. This is a small decapeptide, namely 10 amino acids, very small, which is secreted from the hypothalamus after the hypothalamus detect some external factors. And what are external factors? You probably will probably mention temperature, which is a very important external factor, which stimulates reproduction in some fish. It can be a light, sunlight or artificial light. It is a photoperiod, it's a change of the length of the day, not so relevant around the equator, but very relevant where you go further from equator. I'm talking about photoperiod, but it can be pheromones. Remember pheromones, one fish to other fish. And I'm giving you now example, even common carp. Common carp, when you hold it in a pond, plastic pond, males and females together, usually they will not mate, produce eggs. But as long as you open fresh water with oxygen and even add some substrate, brushes, roots of kind of plants, the carps will spawn, okay? So these are also external factors that stimulate, okay, the hypothalamus to decrease the GnRH, okay? When we know each fish, what are the external factors which do the job or stimulate all these axes, we can sometimes use only external factors to spawn the fish. No need to inject hormones and intervene with all this hormonal system. So remember external factors. Later on, we shall come back to it. I said that the hypothalamus secrete GnRH to stimulate the pituitary. Now look, usually in human being, in mammals, the hypothalamus is not connected directly with the pituitary, but in fish there are nerves that connect directly hypothalamus to pituitary. So if the hypothalamus secretes GnRH, it goes on the nerve cell to stimulate the pituitaries to do what? Sorry, to secrete GTH. Leave this red grief. In a minute, I will come back to it. GTH is gonadotropin hormone. This is a very important hormone that's secreted from the pituitary, remember, or the hypophysis, into the blood system and stimulates the gonad to do some things, okay? So look at the axis here. External factors, GnRH, GTH. By the way, in human being, you probably know GTH is what we call LH or FSH, follicle stimulating hormone or luteinizing hormone. These are hormones which also very important in mammals, in human being. And many times, if a lady cannot come into pregnancy, and the problem is the hormone system, so she's injected with this kind of hormones to proceed and to stimulate the gonad to produce and secrete the oocytes, eggs. Okay, now look what's happening here, and it's very, very important. Grief, dopamine. There's another chemical, dopamine, which is secreted from the brain, from the hypothalamus, but the four letters GRIF, gonadotropin releasing inhibiting factor. That means that we have kind of balance. Whenever the hypothalamus secretes grief, no reproduction. Whenever the grief is low and the GnRH is high, so we might get spawning or response of the female or even the male to our program of spawning. Example. In my thesis, when I was young, beautiful and student, I've tried to see what is the level of dopamine in tilapia at different temperatures, and it's happened we happen to find that the inhibition of dopamine is very high when the temperature is low. The inhibition of dopamine decreases when temperature is high, and then GnRH stimulates the pituitary, and this process is going on. So remember, sometimes we have chemicals which inhibit the process of spawning, reproduction, or intervene with all these axes of hypothalamus-gonad. Okay, questions later on. I was speaking about dopamine and wrote here something. The dopamine inhibition in some intercellular process related to the release of gonadotropin. There is kind of a negative feedback. I will not go into it, but there are chemicals, reverse, there are chemicals which blocks the dopamine activity. You can inject some chemicals and block the dopamine, so you help the fish to do the spawning. A good example is common carp. The inhibition of dopamine is very pronounced, very, very high. And if you inject some hormones and not using the inhibition of dopamine, you will not get female to spawn. But if you inject, and I will show you later, if you inject hormone plus inhibition of the inhibitor, you will get spawning. In this moment, I want to say some fish has a very high inhibition of dopamine, common carp, even catfish. But other fish, marine species, tilapia, even Nile perch, the inhibition is not that profound, okay, and not necessary to inject these inhibitors, which I have mentioned now, okay. Just to give you some example, there's a chemical named reserpine, dopamine antagonist, okay, who prevents the secretion of dopamine from the brain. Another chemical which are very, very in common use around fish breeders are metoclopramide, domperidone, okay, and they are compete with dopamine in the receptor. So dopamine will not have effect. I hope you got it, but again, just to remind you, GnRH in one side stimulate, dopamine in the other side inhibit, and we are trying to play with the levels in order to get spawning. Let's go a little further. Now, what is GTH? GTH is a protein, glycoprotein, namely a protein which covers with groups of sugar, okay? As I said before, it's synthesized, accumulate, and secreted from the pituitary, okay? There are two main GTHs which we are familiar with in human being and in fish. In the past, we used to think that there's only one, but there are two GTAs. Number one, okay, controlling vitalogenesis. In a minute, I will speak about vitalogenesis. This is parallel to FSH in human being. And GTH number 2 is controlling the maturation and ovulation for spawning in fish. Okay, so we have two different GTHs which are secreted at different times of the season in fish. And not only in fish, also in human being and so on and so on. Okay, just Ken.
Before you continue, maybe here there is a question about dopamine. If you can a bit more or again explain what is actually dopamine.
Okay. Okay. Dopamine is chemical which is very important in life of any creature. You know, it's a neurotransmitter in the brain, okay, related to many diseases like Parkinson, many problems, okay. It has been found that if a level of dopamine is high in the blood, sorry, in the brain of fish, okay, it inhibits process of reproduction. Tell you a story, how was it found first time? This was a Canadian scientist who succeeded to physically, yeah, the connection between brain and pituitary. And to his surprise, after cutting the fish, the female goldfish succeeded to spawn. Then he knew something there inhibit reproduction. And then it was the first step to find what inhibit reproduction. By the way, in a minute I will show you, but dopamine plays a very important role in the process of hormonal control of reproduction, because if you don't have inhibition, so a system can come into overflow, produce too much hormone, and this is too much stimulation is never good. So it can play kind of feedback. In a minute I will describe. But for now, please remember, dopamine inhibits reproduction. And I said, if we want to eliminate this inhibition, we can inject here antagonist like domperidone. You don't need to remember, it will be written. But what important is how this experiment is done. You inject GnRH of the brain, and you inject this domperidone. If you inject them both, okay, you see a very high level of GTH from pituitary. If you don't inject the domperidone, so the level is lower. From here we know that if we inject one of the, remember, where was it? Yeah, one of the dopamine antagonists, we simply block the inhibition. Much easier to spawn fish if we inject this inhibition of inhibitor. Sorry, it's a little complicated first time, but by the way, each one of you has the same system here. Each one of you secrete GnRH and GTH and has dopamine, okay? Don't try to play with it too complicated in practice, okay? Now, don't want to go into too many details, but look here on the left side for me, I think for you also left side, there is a secretion of FSH, this hormone which secreted from the pituitary, okay, which goes directly to the gonad. In a minute, I will show you what is it doing there. And on the other side, we have LH, again, one gonadotropin we secreted from the same pituitary, different cells, and this one is so important in spawning, okay? So there are two hormones, I will try to make it a little easier. And now let's see what is vitalogenesis. Remember FSH, follicle stimulating hormone. This is a hormone which prepares the female, prepare its ovary, okay? And now we should see how. So as I said, external factors, GnRH secreted from hypothalamus, stimulates the pituitary to secrete GTH number 1, FSH. This is done usually immediately after spawning. If fishes spawn, the ovary is so-called empty, very, very small, okay? And now they start a process which we call vitalogenesis. And this vitalogenesis is stimulated by this FSH hormone or GTH number 1. What happened? The FSH, so the GTH number 1, stimulates the gonad of a female. Now I'm talking about only females. Till now it was about similar. To secrete estradiol or estradiol, okay? This hormone is detected by the liver, another organ which takes place. And in the liver, there is a synthesis of a very important protein. That's why it's called vitelloprotein, vitel from life, yeah? You all know what is vitelloprotein. If you take an egg of, I will not break here egg, but the yellow part, okay, of an egg is a very high, very rich protein, namely vitelloprotein. In fish, when you look at the embryos, usually there is a yolk sac. This is a vitellogenin. So when female prepare herself for spawning or development of the gonad, the liver secrete this vitelloprotein, which introduce into the oocyte, into the gonad, into the future eggs. By the way, it can penetrate back to the gonad only when we have this FSH in the blood system of the fish. So this is a cycle which we call vitellogenesis. External factors, brain secrete GnRH, stimulates the pituitary to synthesize and secrete GTH number 1 FSH, which stimulates the gonad to secrete estradiol or estradiol, okay, and then the liver starts to, as a factory for protein, to produce this very, very important protein. This protein introduced into the gonad, into each oocyte. Oocyte is the future egg, okay? Egg in its development. And then we see that slowly, the female ovary become bigger and heavier, okay? We call it GSI. I think I have just a minute. No, yeah. I think I have, this is called GSI, Gonadosomatic Index. What is GSI? When you take a fish, you cut its belly, you take the ovary, put on a scale, let's say 200 grams, okay? All the fish was 1 kilogram, so the GSI is 20%, 0.2, namely the ratio between weight of gonad and the weight of the fish itself. And the GSI can predict in some fish whether the fish will spawn or will not spawn. Usually, if I'm going to fish with you, I'm not talking about now about tilapia. Forget about it. It's relevant, but not about tilapia, but catfish. You all probably saw all the carp, silver carp, all the cyprinids. GSI before spawning can be something between 15 and 20%. Namely, if the fish is 5 kilograms, it's carrying 1 kilogram of ovary. And 1 kilogram of ovary can be something like 500,000 eggs. There are numbers which, if you want, we shall give you later on. But if the GSI is only 2%, that means that the fish is not ready to spawn, the oocyte not developed yet, okay? And no one will expect such a female to spawn. So again, before I'm leaving this slide, vitellogenesis is a process which happens after spawning, and this process is simply accumulation of very important protein in the ovary of a female in order to supply in the future food for the newly born larvae, small fingerlings. I will show you some graphs which are related to carps in Israel, but just I want you to see the relations. Look, when you see the temperature on top, where's the arrow here? You see temperature in Israel, okay? January, February, March, Spawning season of carps in Israel is something like April. April is here when the temperature can be 20 centigrade. Okay? Look what happened to the GSI. After spawning, the GSI here in the third, I always put my finger, but you cannot see it. In the third graph, you see a GSI curve. Look here in June, July is 0 because the ovary is negligible, nothing. At this point, the vitalogenesis start, proteins start to accumulate, and the level of the weight of the ovary compared to the body of the fish start to increase. And this is till, let's say, winter, when there is a hibernation. But look what's happening in before spawning. Again, the ovary become bigger and bigger and bigger. And this is related also to a process which we call maturation. Maturation, in a minute I will describe it again, but maturation is the last process before spawning, where the follicle start to accumulate water, to grow and to be bigger and bigger and bigger. And that's happened till the maximum. You see here, March, April, yeah? March, April here, where the GSI is maximum, and this decline of GSI is coming after spawning, and then, of course, the level of GSI is lower. Of course, this is a collection from many, many females. Some have spawned, some not yet. Okay, not more about these graphs. Now I want to come again.
Sorry.
No problem.
Are you going to speak a bit about the vitalogenesis in tilapia?
No, because it's exactly the same, but it's done much quicker, since tilapia is always usually in a preferable temperature for spawning, especially in Africa, or most of Africa, not all Africa. And this process is always happens the same hormones, so on, but the timing is totally different. And of course, if you open tilapia over, you see generations of, at least 2 generations of eggs or oocytes, okay? But it's exactly the same. Sorry, I will say, but thank you for mentioning it, something which is very important to any fish breeder. Vitalogenesis is a process where the fish, I'm talking about the female now, need the best conditions. High oxygen level, water quality, good food, so otherwise we'll not be able to produce this important protein, okay? So usually we, in our hatchery, after spawning, we start in September, October, November in Israel, we give the fish the best conditions so they will be easily ready to produce and accumulate this vitaloprotein. About tilapia, next week, but again, we never force them to spawn cycle after cycle after cycle. Usually after 2 cycles, maximum 3, we put them aside to how can I say, rehabilitation, yeah? A period where they will not be dealing with spawning and only take care of their own body and on this process of vitalogenesis. So just remember, it's not that if you culture fish, you just inject hormone and you get results. Never. You have to prepare the fish, and the female has to be ready, okay, with a proper level of vitaloprotein and receptors and so on and so on. And to inject the hormones, it's only the last step. So where was I? Okay, now I'm coming to a very important sequence of events which we all play while spawning fish. As I said, external factors, and here in Israel, it's done in catfish and carps and many other fish, usually in spring before summer. Some marine species like gray mullet in the ocean, it happens in August, September. The factors are usually temperature and photoperiod. And whenever the external factors stimulate the hypothalamus to secrete the GnRH, yeah, level of grief come down, no inhibition of dopamine. And then we have the process of maturation. I will go to step to show you what is maturation. Okay, usually the oocyte look like this, round organ. In the middle we have a dot. This dot we call it germinal vesicle or nucleus of the eggs, of the egg. This organ which contains the genetic material. Okay, sorry. There are stages. And in maturation, in a minute I will show you how, but you can detect the germinal vesicle or the nucleus migrate to the peripherum. You see, in the middle, a side, in the peripheral, and suddenly there is a breakdown, disappear. The germinal vesicle disappear. All the genetic material is in the cytoplasm of the oocyte. We call it GVBD, germinal vesicle breakdown. Now, okay, and then after it, you see the ovulation. In a minute, I will describe exactly what is ovulation. So, back to this graph. Maturation, as I said, migration of germinal vesicle and also accumulation of water. The eggs become much, much bigger. This is maturation. What is ovulation? Ovulation is when within the gonad, the follicle wrapped down and all the oocytes, the future egg, are concentrated in the middle of the ovary. This is ovulation. And this is happened only after nucleus breakdown. What is spawning? Spawning, and when all this batch of eggs secreted to the environment, into the water. So you see, maturation is a preparation before ovulation. Ovulation is when the eggs are collected within this bag, which we call ovary. It's already eggs, not oocytes, and spawning when all these eggs secreted outside to the water, to the environment. Okay? So again, these three slides represent maturation. This slide breakdown is before ovulation, and ovulated eggs, no nucleus at all. And only ovulated egg can be spawned. Never you will see eggs which were spawned which contain nucleus, never. Okay, now, this might be too complicated, let's leave it. And now I'm showing this process again at a different point of view, which is very important. Look, I was speaking about vitalogenesis before. Look here, from a very, very tiny oocyte, vitalogenesis is accumulation of protein. Look here, the germinal vesicle or nucleus always in the middle. The egg or the oocyte is becoming bigger and bigger and bigger, contain protein, till a certain stage. In maturation, again, look here, I hope you can see the arrow, nucleus in the middle, periphery migrating, and when it disappears, as we said, germinal vesicle breakdown, then the eggs are ovulated. Who found my arrow? Evan, you have it? Oh, here. And then when we don't see any more the nucleus, okay, you see there's still kind of membrane around. This is a follicle. Ovulation, no more follicle, and the eggs are ovulated. So you see here what is GTH number 1 is doing, what is GTH number 2 is doing. Now, practical advice, and I'm always doing it. When I'm taking a female and I want to see if this female will respond to my injection, because if a female is not well prepared, you can inject all the hormones in the world, will not react, will not give eggs. So we can take a sample of eggs using a small polyethylene pipe or glass, and from the opening, from the sex opening, we can take some eggs out and put it in a Sera solution. Look here, it's written here in blue, Sera solution, okay? And this is comprised of 60% ethanol, 30% formaldehyde, and 10% acetic acid. Or giving you numbers, let's say 6 ml of ethanol, 3 ml formaldehyde, and 10 ml of acetic acid. When you mix it, the smell is not so good, I warn you. You mix it and you take a sample of eggs from the female and put inside. After 3 minutes or so, the protein of the egg become clear, and that's a picture which you see, okay? Membrane and nucleus. Whenever you see that all the eggs are in a central position, no chance. What's happened here? Sorry. Yeah. No chance that this female will spawn. Whenever you see that most of the eggs, let's say 60% and more, are in peripheral condition, like you see here, the cells from the right, yeah? When the nucleus is not in the middle, that means that the female is in a maturation process and will probably respond to our injection. So it's a very common process or protocol to take sample of eggs, put in this Sera solution, and see or try to predict whether this female will or will not spawn. Giving you example from my hatchery. Once we had a very tough competition, who will supply carp to the market, fingerlings to the market. So usually carps spawn here in April. I wanted to spawn them in January in order to be the first one and get all the market. So I brought the fish from the field to the hatchery. I took a sample of eggs, and all the eggs were in a central position of the German vesicle. I knew that when I will spawn, I will inject, no female will spawn. So what we did, we have started to heat the water slowly from 14 or 15, I don't remember, till 33, very, very slowly. It took us 1 week. And after 3 weeks, we've started to see that eggs become in a peripheral position, okay? And then at the end of February, we have succeeded to spawn the fish. Without this technology of sampling ovary and looking at eggs, I couldn't spawn the fish. I would inject hormones and nothing would happen, okay? So the picture which you see here is, on the left, at least in my position, GTH estradiol is vitalogenesis, and then maturation, the hormone which controls this process, and what we are doing in order to know if the female is ready or not. Just a minute. I will take water. And let's go on.
Achik,
כן.
מרטינה.
שאלות אפשר, זמן רץ. בואי שאלות אם רוצים.
Sorry?
אם יש שאלות, no problem.
Yeah, I think because it's just about the subject you were talking. So Martina is asking, I don't know, I don't remember from where Martina is. Martina, would you like to ask by yourself, maybe? So we hear your voice.
Yes, Martina.
Yeah, hello. Nice to meet you, and hello to everybody. I follow with great interest your speech. However, I have a little bit of doubt that what you explain to our colleagues in Zambia and Madagascar and Malawi can be really applied in the countries. I understand that it is very important to understand the background and the methodologies, but in how far can a fish farmer in Malawi apply your techniques?
Great. Okay. Look, Martina, I know that. Also in Israel, you go to most of the farmers when they see this graph, they'll say Chinese, my Chinese, we don't need it. We know how to produce fish. And truly, it's only to give background. Of course, any farmer can do most of the work without be aware even to the hormones we're sitting behind. But, and that's my point of view, people are doing what they learn to do and what is the most relevant to their conditions. And no doubt the farmer in Zambia will do the best, almost the best, for his conditions. But things are changing so much, and sometimes arising problems, and you don't even have a way how to judge it, where to look for solutions. Let's say you have in Zambia this very small sardine. How does it call it? It's a very, very small fish. Most important, okay? And people don't know how to spawn it in the hatcheries. Capenda or something like this, am I correct? Never mind.
Yes.
Somebody can help?
Never
Yes,
mind.
Scapenta.
How Masuto?
Scapenta.
Capenta, okay. Not many people knows how to do it commercially, how to produce. It's very important fish in the Zambian market, even though it's very, very cheap, very, very small. So, but if someone wants to solve these problems, he has no chance but to know what's happened, where to look for. Maybe yes, to use hormones, maybe not hormones, how to play with external conditions, things like this, okay? So you're right, these farmers which raise tilapia, which he finds in the river, throw in his home and put some cow dung, does not need to know it, okay? So what I suggest, remember it in your brain. You don't have to use it, okay? If it's really not important, even forget it, okay? It's only 1 hour of your life, okay? But whoever will cope with the problem of fish with somehow not spawning, or from any reason the level of fertilization is low, he has to go back to theory. No other chance to get progress. So to be more rude, and I don't want to be more rude, you don't need me if you want to do the same as you do now, okay? I'm just focusing on future progress or problems which will arise. That's all. Okay, I will, but Martina, 10 minutes and I'm finishing. So, sorry, another question?
No, you can go on, Achik.
Okay, so now I just want to show you how myself, I'm using it in a hatchery, all this theoretical background which I have described, and I call it level of intervention in this spawning. So the first level, and I am for it, is using external conditions. If you can use only external conditions to stimulate fish to spawn, that's the best. Cichlids, all kind of cichlids, all the orechromis, all the tilapians, and blah, 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 no need to be injected, since whenever you supply them good conditions, optimal temperatures, and of course the body is healthy, they will do it by themselves. But let's say about myself, I'm producing goldfish, millions every year. So sometimes I'm taking the female, put the cannula, take eggs, see how, blah, 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 and do science and inject. But lately, we have paid attention that when we supply them with brushes, okay, and a proper temperature, they will do it by themselves. So only using external conditions, manipulations, if we get the eggs, that will be the best and most healthy for the fish. And more than that, let's say goldfish. If you inject them with hormones, they will do it only once a year. Because you intervene in all this system. It's very rude, yeah? It's like taking a hammer on and hit the head of the fish. But if you can cause them to spawn using only manipulation of external conditions of environment, they can do it even 3 times a year. So that's preferable. But unfortunately, many fish you cannot play. And I give you an example. We have this fish in Israel, you have some of them in Africa. Many fish which came from Asia, China, okay? Fish for ecological purposes. Fish which was introduced to eliminate snails for bilhartia or to eliminate plankton. All these fish will not spawn in Africa. I'm talking about some fish, maybe you are not aware to, silver carp, grass carp, black carp, never mind. This fish, you do whatever, you can do whatever you want, but you cannot produce big rivers, monsoons, you cannot change the condition. And then in this fish, no choice, you have to inject them with hormones. Okay? Usually, playing with external factors is good in small tanks, aquariums, hatcheries. In the wild, it's quite problematic. But the first level to induce fish to spawn will be playing with the external factors. Second level, okay, I wrote what are the external factors. Second level, and this is, sorry. Second level is to stimulate the pituitary using this GnRH. And this is all over the world. It's the most common system now, okay, to introduce, to cause female to spawn. And then whenever you inject the GnRH, you stimulate the pituitary, yeah, to secrete its own GTH. Okay? And when you do it, you can use also the dopamine antagonist, as written here, domperidone, metoclopramide. No, not important. I will not go to which is most advisable and so on. If someone really wants to do, I will privately give him some data. And the old system, which is still used, is simply injecting GTH. which stimulate directly the gonad. And you have to understand, if you do it, the gonads have to be well prepared. So what is GTH? It can be, just a minute, I'm going to bring you hormones from my fridge. Sorry for the, where is it coming? Okay. I think you can see here the very, very small balls, white balls. These are pituitaries of common carp. Each one of you has one pituitary in the head. Okay, also I have one. These glands are GTH because they contain GTH, which was produced, okay, and collected from brains of fish. It can be HCG. You see here, HCG. HCG is human chorionic gonadotropin. This is a hormone which is produced by a pregnant lady, human being, okay? And it's very similar to HCG of, GTH of common carp. By the way, in where I live, in my kibbutz, okay, in Israel, the first fish breeders, I was still a sperm at that time, okay? The first breeders used to collect urine of a pregnant lady, inject it to the carp, and they got spawning because the hormone is quite similar, the GTH of carp and HCG. And PMSG, pregnant mares, serum gonadotropin is from a female horse. Now, when you inject this GTH, you override all the brain, you override pituitary direct on the gonad, and the gonad will respond. Now I want to give you some thoughts. What is more advisable, okay? One, the GnRH is, as I said, very small molecule, and it's the same molecule plus minus in all the fish. So you know that you can take, and this is synthetic produced, okay? So you can take this small molecule, generate, and inject it to each fish, okay? Will respond. More than that, because it is very small, it does not create any antibodies in the body of the female recipients. But if you inject GTH, the big hormone, to the gonad, and sometimes, because it's a strange molecule, after 2 or 3 times, it might stimulate the body to produce antibodies, and fish will not react. So whenever possible, I'm using GnRH because it's synthetic, no problem of viruses and all kinds of things, okay? And will never create immune response. But as you can see here, I'm still using pituitaries, and look here why. And this is very important to know. You can see here 4 graphs, sorry, 4 graphs. And this graph describe you a different temperature, okay, the bottom line from 20 to 27, and on the side, okay, I don't know if for you it's the left or the right, in my side left, it's how many hours after injection you can get the eggs. Okay, and you can see that the 2 top lines are GnRH. Why the time is much longer? Because the GnRH stimulates the pituitary, and the pituitary is supposed to secrete its own GTH, and this is supposed to stimulate the gonads. It takes time. Whenever, and if you see the 2 bottom lines, the time is half time, much shorter, because you stimulate directly the gonad, you override all this process of secretion of GTH and stimulating the gonad. You have to be aware to it when you work in the hatchery, because sometimes if you inject fish and you come too late, the eggs are not good anymore. And when you inject GTH, you have to come earlier. When you inject GnRH, it's take more time. But look, all the hormones are much quicker when the temperature increases. Okay, if the temperature is, let's say, 20 centigrade, it might take 21 hours. If it's 26, it will take only 11 hours. So you have to be aware to temperature, of course, to the hormones which you are using, and how to know when the fish is ready to spawn. And this we shall speak later on. This was very long, I'm sorry. So now I gave you a general background, really a general background of what's happening within the fish, okay? Hormones, how we can manipulate, and so on and so on. Please, if you have any questions or remarks, do it now.
Buga, would you like to ask directly?
Yes.
Please.
Yeah. How do you consider age of the fish, maybe size of the fish? If you want to extract the pituitary gland that you want to target the GTH, and if so, how do you process the hormone for storage?
Okay, thank you. Thank you. I will just fix my. Okay, and you're talking about this. Look, this hormone, this pituitary, yeah, is from common carp. It was collected in a factory where they process common carp. They have nothing to do with the heads. In Israel, nobody eats heads, okay? So they cut it and took the pituitary out of the head. I cannot show you now, I will not cut mine, but anyway, it's possible to collect. Now, usually after we collect this gland, okay, which is very, very small, we put it in ethanol. Wash all the oil from the brain, it's become clean. And then you can do two things. One, change the ethanol, wash it, and you can keep it in ethanol even 3 years. I don't know if maybe more, but 3 years I can guarantee. But usually what we are doing, we put it in acetone. Acetone is doing kind of dehydration. Take away all the water and it's become dry. And again, I'm keeping these hormones in my fridge for 3-4 years even, okay? No problems. This hormone, this gland from common carp, I'm using for grass carp, black carp, catfish, okay? But sometimes I have fish which I cannot use this gland. I'm using from its own species, okay? If you want to special fish, you collect glands, and you have to know when to collect in order to get the higher level of hormone within the gland. So usually it's little time before spawning, okay? Little time. Look, when we inject these glands, in the next lecture I will show you, give you doses. We always give too much. It's not calibrated on milli on nanograms, okay? So even if you don't inject, or if you don't collect in the right season, usually I don't see any problems of glands. We collect from both males and females together. Yeah, we do not separate because it's the same hormone, same hormone. So again, to your questions, if you know how to collect, you put it in ethanol. You can use this ethanol even, look here. Finlandia, but high level. I'm kidding. Any ethanol or dry it in acetone. This will be the process. Okay, have I answered?
Yes, you have answered the question.
Thank you, Achik. Thank you, Buga. Coming back a bit to vitellogenesis, Phiri Hazina, would you like to ask, please, your question?
Where from?
Vitsi is from
Africa.
This is complicated. We have a lot of people here. It's a big group.
Okay, never mind. She will
I read the question.
Okay.
In common carp, what diet do you recommend for a brood mare to have a normal vitellogenesis process until ovulation?
Excellent question. It's quite easy. First of all, I have to say, it's not only the food. Conditions, for me, is much more important. I'm 100% sure that Dr. Bejarano has mentioned you that you have to know how to grow water, not to grow fish. The fish will do by themselves. So we keep the broodstock in a low density. In our condition, we call it low density. And low density can be something like 2 tons per hectare, females, not more. We regard it as low, sorry. We give them 35% protein pellets. Part of the pellets contains fish meal. We taking care of oxygen, namely if we have problems always aerating or introducing fresh water, trying not to expose the fish to oxygen depletion. It's a terrible thing in aquaculture. And that's all. Okay, in our temperatures, we're giving something like 1% in the winter, 0.5% per day of body weight of the food. But please, don't take my word as a formula to your conditions, never. Okay? But just remember, high protein, we call it 35%. Now, if you have body water with natural food, if it's Daphnia, Moina, whatever, so you have enough protein probably, then you have to add, you can use less protein and high carbohydrate food. And believe me, it's so easy, yeah? These fish are not spoiled. By the way, one remark, you know, carp in Africa, it's not so popular. But in some places which I have visited and worked, we didn't have chance because there is winter and sometimes water temperature drop down to 14 centigrade, 12, 15, and tilapia is not a good candidate. And they've succeeded to produce carp, and after convincing the market to get it, okay, it's excellent candidate. Okay, one more question.
I think we can continue, Achik, and
Okay, so now look, we should go now to practice. I mean, so-called practice in Zoom. And I will try to relate my work to the previous lecture and fish spawning method and technology and technique. Something wrong here. Why? Why is that? Because maybe that's why. Yeah. Remember, I will not go into it too much, but just to remind you, external factor, generate, GTH, spawning. Okay, Now, advices from me. There are certain systems of spawning fish. First one is natural spawning. You take mommy and you take puppy, okay? You put them in the bed, you put the curtains, they will do it by themselves, believe me. I think so. I know not too much experience, even though I have 3 grandkids. In suitable condition, timing, fish will do it spontaneously. About tilapia, next week we should think they do it spontaneously, but also carps. Okay? Now, there is in a commercial farm, sometimes you want to synchronize all the females at the same time. You don't want, even though they will spawn, you don't want female to spawn today, another one to spawn next week. There will be a difference in size and things. So we do a natural spawning, but we inject hormone. So now we calibrate all the females, plus, minus, to spawn at the same time. For this process, we need more males. Okay, just imagine 10 females, so we need maybe, not maybe, at least 20 males to do it. And I will describe it in a minute, but just to give you the idea, even those fish spawn naturally in the pond, sometimes we want to inject them to synchronize all the females at the same time. And the last one is induced spawning or stripping, doing in a hatchery, taking the eggs. I will describe it in a minute. Okay? Now, in this system, usually we get much higher fertilization. When I'm spawning carps or goldfish in my hands, okay, I get something like 90–95% fertilization rate. Most of the eggs are good. In a natural spawning, if you get 60%, lucky you are. It's good, okay? Now, another thing. Sometimes, for many reasons, we want to do hybridizations, and fish will not mate on each other. Even in the tilapia or chromis, sometimes when we want to produce hybrids for many reasons, so we not do it by themselves. Courtship is different, okay? So you need to do it with your hands, okay? Otherwise, they will not. And as I described before, out of season. Out of season, naturally, no fish will spawn. That's the reasons where we are doing induced spawning or stripping. And now advice, which is very important from my point of view, and I know it's not practiced in Africa. Look here, it's written under tranquilizing with phenoxyethanol 60 to 100 ppm. I will go back to it later, but please remember, now in my farm in Israel, any handling of fish, if it's for spawning, if it's even transportation fish which are very active, we put some tranquilizing. It's a chemical which calms down the fish, Fish are not active. They consume less oxygen, okay? And no problem after the period when you take them out of this water with this chemical, you put them back to fresh water, come back to activity, and so on and so on, okay? So I know it's not so practiced in Africa, no reason. I'm giving you an example. I was working in Azerbaijan producing caviar. Caviar is very precious eggs. And we had to spawn females of 49 kilograms, something huge, okay? Now in Israel, we would have put her in the tank, put some chemicals, the fish would come down, no problem to collect the eggs, the female is not getting wounds, does not lose scales, not losing mucus. Very good. They didn't know the system and started to hit it with wooden blocks just to calm her down, things like this. So any handling now, I recommend to use tranquilizing materials. Here it's written one of them. I will come back to it later on. Go back to natural spawning. Always we need spawning ponds. This is relevant also for tilapia. Pond has to be dry and drainable before spawning. Why? Just imagine, you know, tilapia maybe or rehomis, whatever. If there are small fingerlings of fish or larvae in the pond and you put the broodstock, they will spawn maybe 7 days later, okay? You should have a variation in size. The big fish will eat the small fish and so on and so on. So, dry ponds, okay? And drainable, this is also a good way to collect the small fish in an easy way and so on. Now, for carps or many other fish which have eggs which are adhesive, I think most of the fish simply attach the eggs to substrate, okay? So we supply with artificial substrate. It can be only pine tree. Pine tree, you know, has leaves, okay, which last a long time in the water. It can be roots of water lattice. How does it call in Africa? It's very popular, okay, never mind. Nylon brushes, whatever, okay? To each female of 5 kilograms, we supply this spawning area of 10 square meters, plus minus. Now, giving you numbers now. Female of 5 kilograms, something like this, can give us 1 kilogram of eggs, and this is something like 600,000, little over 500,000 eggs. That's why we need big area, because if you don't supply with big area, all the eggs will attach one to each other. It will be like big omelette, and eggs will not get enough oxygen during spawning, during incubation, sorry, okay? So we supply them with area, and the eggs are separated, attached to the substrate, and get enough oxygen. Fresh water, always good. And look, here's the ratio. 2 females and 3 males in each pond when you want them to do it by themselves. When I spawn in the hatchery, in a minute I will show you, I take 2 males, and this is good for all the season, no problem at all. Very briefly, this is a spawning pond, okay? Spawning area in the deep part. In the deep part of the pond, we can introduce fresh water there, never mind. This is a drawing, very old drawing from Hungary, of how it looks exactly, how the eggs are attached to the grass, okay? Show you some, now, I was talking about tranquilizing the fish. Here you have the doses of MS222, not using anymore, it's not health. Quinal D, not using anymore. Phenoxyethanol, this is the best stuff. And clove oil, these chemicals knock down the fish. If you put, instead of 100 ppm, only 10 ppm, the fish will be quiet. If you want the female to lie down, okay, we call it, how do we call it? Rape drug, yeah, some honest. The female is sleeping, and then you can collect all the eggs or transfer her from place to place. Nothing happened to it. Remember, there are chemicals which are very good for handling fish. These are the chemicals. You will see later on some experiments about the levels of these chemicals to calm down the fish. And not only spawning, even tilapia. In Israel, some people, when they transfer fingerlings of tilapia a long distance, in the tank they put this tranquilizing, and the fish are not striking one each other. They are very calm. The oxygen consumption is lower, no problem, and the fish get to the target ponds in a much better shape. And of course, the survival rate is much better after using this. But you have to have practice how to do it, and so on and so on. Now, if you remember, I was talking before about 2 levels of intervention. One is injection of GnRH to stimulate the pituitary, and the other one is to stimulate the gonad. Look here. Here you have the protocol of spawning. In dark blue, this is the bottom, okay? This is a GnRH, the synthetic hormone, enough to give 1 injection. Minus 14 hours, that if you want the fish to spawn, let's say 8 o'clock in the morning, you take 14 hours reverse, you give the injection, and you expect to get it plus minus 14 hours after stimulation injection. This is when you're using GnRH, very popular. Not this one, not the glands, GnRH. I have also, but it's powder, so nothing to see. Now let's go to using pituitaries. And now, why am I emphasizing? Because a farmer in Africa wants to induce his catfish to spawn. Hard for him to find this synthetic hormone and so on and so on. So he can take from the river or collect pituitaries and put in the ethanol in the fridge, okay? So now this is relevant, this protocol is relevant to carps, catfish, and many other species. We give 2 injections. The first one is called the priming. Okay, we give it 24 hours before spawning. And the doses is written here, 0.2 pituitary to kilogram. That means, I want to open and not break it, just a minute. Yes. Yeah. Where's the camera? Here. 1 gland like this is good for 5 kilograms female. Minus 24 hours. After 12 hours or so, you give the second injection to the same fish. And the level now, or the dose, is 5 times more. 1 gland to kilogram of a female. Okay? Now, it's very important to look at the temperature. When temperature is cold, time is longer. When temperature is warm, it goes everything much quicker. So look here, difference between the 2 hormones. Pituitary or GTH, 2 injections. GnRH, the synthetic one, only 1 injection. Okay? And if you remember before, the time till fish will spawn will be much longer using the GnRH, the dark blue hormone GnRH. And if you're using, by the way, I didn't say, CP is a carp pituitary extract, okay? It takes less time. I did not say that you cannot inject this gland to the fish. I did not bring it here, maybe next lecture. We have kind of homogenizer or a coffee grinder where you collect the glands, make it kind of powder with some water and inject. You can use distilled water. You can use saline water. You can also use Coca-Cola. It's not important because the the volume is so small. So usually I'm using saline water from this, bags, but you can use tap water also or distilled water. Not important because such a big fish will get 0.5 ml of any solution with this hormone will be good. This is not an issue at all. Where to inject? Not important. Injection can be in the muscle of a fish, can be interperitoneal into the body cavity, not important. You just have to make sure that you don't hit the heart or any important organs, and it's very, very important. Okay? Now here, there are very old drawing I had to show you how to introduce fish to. Here, injection in how to produce fingerlings in a very simple system. So, where again, here the arrow here, you can see that they're bringing the fish into the field, inject them and release them to the pond. The fish will do it by themselves, okay? Possible to do it in cages. Very practical also in tilapia in Africa. We should talk about it next week. In cages, you can do anything. Everything is under control. You can easily collect the eggs, and later on collect the larvae, and so on and so on. Okay? And you can also here spawn with your hands. Stripe it, okay? And attach the eggs to I think it's called in Africa kakaban, no, all kinds of roots or brushes or whatever for incubation. So these are very simple system, sorry, to produce fingerlings. I have 20 minutes, and now I would like to describe you last time about the hatchery process. I will do it very quickly. In artificial induced spawning in a hatchery, usually we have very carefully to manage our broodstock. It's never in the hatchery, it's always in the field. So back to the question which I was asked before, fish has to be very well managed. Feeding, conditions, and so on. Before you bring fish to the hatchery from the field, you have to disinfect the broodstock because the broodstock can carry all kinds of parasites. And if you bring parasite to the hatchery, one more problem which you have to cope with. So probably Bejarano was speaking, we usually disinfect with formaldehyde, 25 ppm, 6 hours. I hope Bejarano has talking about it. Anyway, to disinfect. If somebody does not have all these formaldehyde chemicals, a very good solution to disinfect fish is salt, table salt. Very high level, ocean level, 30 ppt, 3 minutes, it will kill all the parasites, and fish will suffer a bit, but can stand it. Okay, and then we give first injection, remember? First injection, 24 hours before. We call it priming. Look here, number 3. And then we do give the injection after, induction after 10 hours. And then we are stripping males and females. In a minute, I will show you a video film from my hatchery. You will understand. We collect the eggs. We put the sperm on the eggs. And then we fertilize the eggs. Listen, sperm of fish is not active unless you put it in water with a low salinity. Okay, so if you take bowl of eggs and you take from the male sperm and you mix it, not enough. sperm is not active. You have to add fresh water, and then you stir it, mix it very well, and only then the sperm become active and fertilize, penetrate to the egg. Okay? Now, the sperm has very short lifetime, 2-3 minutes, not more than that. That's males, a lot of noise for short time. Again, the sperm, same. So after fertilization, you have to remove adhesive material from eggs of common carp because they will attach one to each other, and again, you get omelette. No good. So how to separate in a minute, I will show you. Okay? After you've separated the eggs one from each other using some chemicals element I will describe, then you can put it to incubation. Okay, these are the steps. All the steps are here. I don't want to describe since we don't have time. Look here, this is incubation in my hatchery of carps. And you can see the larvae, and under the larvae you see the larvae is not pregnant. It's carry the yolk sac. Remember, vitellogenesis. In tilapia, it's even more clear, okay? You see the yolk sac, which supplies the food for the first 2 days, 3 days, it's up to the species and the temperature, for the first stage of. Now, here I describe, I do very quickly, how we separate, how we remove the adhesive material from the eggs so they will be separated. I don't talk, everything is written, but what we are doing now is we are using milk. We put all the fertilized eggs in milk. It's written here, milk plus water. You put inside and you mix it or put air stone. What is the milk doing? The milk attach, the particles attach to each egg, and then they will not attach one to each other. That's the easiest, the simplest way. In some places where you don't have milk, you go to the river, you take silt, mud, you put inside, and the mud will cover the egg. There will not be problem of oxygen. Don't worry, okay? And this has very simple systems to override the problem of adhesive materials, okay? Number 2 and 3, you will read it. Some chemicals which you simply remove the adhesive material, okay? So these are anti-adhesive systems for incubation. And now here you see a picture of development of the embryo, and it's so nice to see it under microscope, okay? Not important the names, mola, blastula, blah, 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 just to see. Here you see a fertilized eggs. First division, 2 cells, 4 cells. Here we call it like strawberry creatures. And slowly you see how the embryo developed, okay? And only in this stage, you can assess the level of fertilization. Sometimes we do all the spawning, and for many reasons, all the eggs are dead, okay? So whenever you know this stages, you can assess your success. Usually in common carp, if you want, you can use microscope after 10 or 15 hours. If not, after 20 hours with your eyes, you can see whitish eggs are not good. Clear eggs with the development, which you can see, are good eggs. Okay. Now I don't want to do it too complicated. This is for our next lecture. Okay, Now I want to show you how we are doing it practically in the hatchery, and I will show you some short movies. Sorry, I hope it will be okay. Where was I? Let's go up. I'm sorry. Can you see? Sharing is okay?
Yes, yes.
Okay. First of all, you say we have a very big fish named silver carp or bighead carp. This fish is so active. When you handle it, it will jump to the sky. It will fall on the ground. So always we're trying to calm them down in the small tanks. So let's see if we can. You see how the fish is quiet. Oh, sorry. How the fish is quiet since.
No, we don't.
Sorry.
Wait, Achik. You have to. Maybe you have to open again as a presentation.
Have you seen anything? New share?
No. Share.
Don't tell me.
No.
Huh?
No, you did a new share? No. Now, maybe.
I did a new share.
Okay.
Sorry. Sorry. Just a minute. I'm sorry. I have to apologize.
Slowly. Okay.
Okay. Now I would like to show you how we check if the female, after I put her in a tank, if she's ready to spawn. And let's see if you can see anything.
Yeah, it's okay.
Can?
Yeah.
You see, I'm taking the, not me, myself. In these tanks, we put chemicals and the fish are very quiet. That's how we choose a fish for spawning, okay? Just a minute. Females ready to spawn, female ready to spawn. Have you seen how Lee, my friend, is touching the belly and some eggs are coming out? After coming out, we put it back to the tank, and the female are tranquilized, very quiet. And when we want to collect eggs, I will show you silver carp, how I collect eggs from silver carp. You see it?
Yeah.
You see, simply collect eggs. No, sorry. Okay, just a minute. Something here in my computer is a bit. I have to apologize. Just a minute. Yeah. Okay, now I have silver carps, which I want to induce to spawn. You see the eggs? The female is very quiet, right?
No, wait.
I'm taking eggs.
Achik,
Huh?
you have to do sharing again. No, you have to open it. Sorry.
Yeah. Sorry. Now maybe?
Yeah, that's good.
I will go reverse, show it again, how we do it. Son of a bitch. I have to apologize.
No, not those words.
What? I wanted to. New share. Yes or no?
Yes.
You see how the fish is so quiet? And if I did not do it, probably 50% of the female will die. Okay? So using this tranquilizing is so important. This system is used for all the cyprinids, for even catfish with a small differentiation. I want to show very simply how we fertilize the eggs. It's so simple. New share. You see, eggs, sperm, put simply tap water. Only then sperm start to be motile and fertilize the eggs. That's all the story, very simple. And me, myself, I have built this year a hatchery which is very good for Africa. Very simple. Anyone can do it if he wants. Now, anytime after we collect the eggs, we measure the volume, and then we know how many eggs we have, okay? How many eggs we have. So most of the time in silver carp and carp, it's around 500,000 eggs per liter. 500,000 per liter, okay? Incubation. These are the incubation which we are using. You see, water is coming from below. Eggs are heavier than the water. Where are we here? Eggs are heavier than the water. Yeah, and that's how the eggs get the oxygen. Eggs always have to be in a movement, yeah, in order to get oxygen. And now how it goes, it's very important. As soon as the egg is hatched, a very small creature named larvae is hatched. The larvae has a tendency to come up and take first air, breathe, as somehow to inflate the swim bladder organ of the fish. Because the water is always introduced into the tanks, okay, the larvae is carried out with the water from the incubator into the nursing tank. That's what we are doing usually. Now, Evelyn, I have promised, and I hope to fulfill my promise, that's to give you a full video today or tomorrow. Anyone will be able to see step by step good video which was taken in a hatchery in Israel, and maybe next week, if there will be questions, we shall answer them. But please remember, sometimes it looks complicated. Not at all. Anyone can do it at home. Just use his conditions, okay? And just to know what he's doing. Okay, so I have to apologize for this videos which was not organized so well, but you got the idea how we collect eggs, we put sperm, put water for fertilization, okay, and put in incubation. Now, important to know how much sperm do we put on eggs. And now, boys, you will have to forgive me. Fish usually has much more sperm than we have, human being, yeah? Much more. I've taken from bass once 200 ml from 5 kilograms, okay? So no need to use so much sperm because sperm is so much concentrated. 1 ml of sperm to 1 kilogram is enough. Usually, farmers, they don't even measure. They take the eggs, they put the sperm on the eggs, and that's all. Some advisors, just a minute, I have to go, okay? Okay, some advisors. 1, whenever you deal with fish in the hatchery, you have to make sure that you wipe with a towel. With a towel, you wipe the female in order not to bring any water with the eggs. Why? Because eggs, whenever they feel water, whenever they touch the water, it activates the adhesive material, okay? Or fish with no adhesive material, the eggs start to swallow water and blocked from penetration of sperm into the eggs. So we make sure no water will penetrate to the eggs. Another thing, as I said before, whenever we are collecting sperm or playing with sperm, no water, because water is a trigger for the sperm to start his run, and you have only 2 minutes. So if you put water, after 2 minutes, it is dead sperm and will not fertilize eggs. So if you want to collect sperm in a vial, you wipe the male, collect the sperm, cover it, and can put it in a fridge. This is good for almost 24 hours. Dry, no water. Whenever you want to fertilize, you take the sperm, put on the eggs, of course, mix it, it will heat a bit. When you put the water, sperm will start its motility or its travel for fertilization. Me, myself, whenever I'm playing with males, I collect some sperm, put under microscope, it's all dead. I put a drop of water, and then I can see that the sperm is motile and can be used to fertilize the eggs. So no problem under big magnifying glass or under microscope to see if the male is good. Always we're using more than one male, because sometimes sperm of one male is not good. So to be on the safe side in commercial processes, we take 2 or 3 males for each process. Okay, these were some remarks at the end. Let's give some minutes for questions before we go home.
Yeah, great. Achik, thank you so much. There are some questions here. And please, if there are more questions, raise hands, okay? Maybe we begin with Asafu. Asafu had a question shall I read it, or Asafu, would you like to ask directly?
Yeah, thank you, Evelyn. I wanted to know on the issue of temperature, what is the optimal level of temperature for possibly catfish to spawn after injecting?
Which fish?
And the other question I want.
Catfish. Catfish.
Catfish?
Catfish.
Okay.
Yes. And the second question was on, you said we can mix eggs with the milk or mud from the river. Now, taking mud from the river, aren't we taking pathogens to infect our eggs?
Good.
On the use of chlorine, water.
Yeah, okay.
Tap water normally has chlorine. Won't it have any negative effects on the eggs?
Okay, Asafu, clear. Your remark was very important. I was just kidding, telling you that you can take Coca-Cola, Sprite, whatever. No, what I said, it's not important if the water is tap water or distilled water, or don't take from river, you never know what happened there. Don't take Coca-Cola, of course. Just wanted to show you that it's not that important. And if somebody will tell you, you have to take saline water, and if you don't have, no way, take water from the tap is good one, okay? That's what I meant. Never take from the river, never take from places which you want. Okay, regarding catfish. By the way, I'm very fond of catfish, because this fish is so strong, can be cultured in hard conditions, and you know, some places that's the only way to succeed. Optimal temperature for catfish usually will be from 26 to 30, 28. Okay, not that important. Okay, so me, myself, here in Israel, we don't play with temperatures at all. So we start to grow catfish. It's, by the way, on a very low scale, because no market in Israel. From 26 to 30, and spawning at the same temperatures, no problem at all. Next time, very quickly, I can show you a presentation of catfish, which I have done for Uganda people. But as I said, 26, 28, excellent temperature for any of the catfish which you're dealing with. Of course, in Europe, there's another catfish, Silurus glanis. Temperature is much, much lower, 18, 19, but this is something else. Okay?
Thank you, Achik. Trevor, would you like to tell us a bit about your area and your question? Trevor. Trevor, you are with us? Well, I think, Achik, you spoke about that, but maybe it needs a bit more of explanation. Trevor is asking if fish sperm can be preserved and for how long?
Okay. In fish breeder system, us, not scientists in a lab, if the sperm is not diluted in water, by the way, can be diluted in very high salinity, but let's say not diluted, 24 hours is still good. After that, no recommend. Of course, there are techniques to preserve sperm from ages, for generations, but this is something else, a deep freeze, and you need all kinds of chemicals and so on. We're not talking about it. In the field, if you collect sperm, you put it in the fridge, put a nylon. Don't put it in zero, because if it's freeze, melt, the sperm might be broken. You put it in the 4-5 centigrade. Okay, this is very important. Okay, don't make it ice cream. I don't think it's tasty, never tasted, but it's not tasty and no reason to freeze sperm. So till 24 hours in good conditions, no problem.
Okay, thank you. Well, I see Buga with the raised hand. Buga, please.
Yes, thank you so much. I have a question.
Buga, show yourself. Okay. Oh, so beautiful. Yes. Why not? Yeah.
Yeah, I have a question on catfish and possibly of common carp, because all these fish species, they will need substrate for them to spawn. If you have done the spawning, maybe in tanks where you put your substrate, so after hatching, you may have some kind of eggshell that remains in the tank. So how do you get rid of the water quality with regard to the fry that are emerging from the spawned eggs in this situation?
Good. Okay. You're totally right. You can spawn on substrate in tanks. Yeah, and if you don't replace the water quality will deteriorate due to eggs which are not fertile, eggshells, and so on and so on. Always, we recommend to change the water constantly, okay? At least 2-3 times a day, the volume of the tank will be replaced. That's the best system to ensure high quality water. But if you don't have the facilities, you can do the spawning in tanks and then take all the eggs on the mattress and transfer it to an open pond. And then they will hatch there and you don't have this water quality problems. Other problems will arrive, but you can do it. And I have done it, believe me, in goldfish. I've spawned goldfish on substrate and took them 300 kilometers. 300 kilometers, spraying some water on the way and transfer them to big lakes for incubation and hatching. No problem. So in your farm, if you don't have the facilities, you know, to replace water in tanks and so on, you simply can move it to a bigger volume, bigger pond, big volume body of water, and then usually water quality is less problematic, okay?
Excellent.
This is very relevant, by the way, with catfish as well.
Yes.
Tony Carps.
Thank you, Achik. I think we will go to another 2 questions. Okay. And maybe it would
okay,
be...
let's do like this. 2 more questions, and then you go home for homework, you know, reproduction. And so the next week, tell us what happened. Yeah.
No, we are not. We have still work, but
okay.
no. But what we can do is to collect the questions, because we will be with Achik next week also. So if there are questions, please send them or by WhatsApp or by mail, and so we can answer them maybe at the beginning of next lecture. But
yeah, we are not going anywhere. We are
so
connected.
Here is a question of Lojarano from Madagascar. Let's see if we have Mike. Lojarano, do you have Mike? Do you want to ask by your turn? Yes, please.
Can you hear me? Thank you. You were talking about water heating during cold season, and I would like to know what kind of machine or are you using archaic method to heat water during cold season?
To heat the water?
Yeah.
Okay. Look, I'm always talking about small volumes. Small volumes. In Israel, we have no other choice but using electricity. Okay? So now we had the ambient temperature was something like 15 centigrade. I wanted to get into 23. So every day we have increased in 1 centigrade. We have these thermometers, okay, and using electricity. Water replacement was minor, okay? So it's not affect the process so much, but we did not have any other choice but using electricity. Nowadays, there are other systems like solar systems and things like this, but since the volume of the tanks is so small, the cost of the electricity is not that high, okay? But using electricity only. We have a reliable system here for electricity.
Okay, thank you.
Okay? Anytime.
Let's go to Matthews. Matthews Chait. Yeah, Matthews. Matthews, you are from Zambia. Do you have a mic? Are you okay? Matthew? Okay, I will read the question. Once the eggs are laid, spawned, how long the eggs of tilapia can stay awaiting fertilization so as for them to hatch?
Good. I don't have clear answer for that. I don't have clear answer for that. In nature, you know, it's a matter of seconds, 1 minute, even less. I don't have an answer for what is the maximum time. I will, for next lecture, I will try and get answer. Sorry.
Great. And one question from James. How long should the fish be handled after applying tranquilizers?
How long?
How long should the fish be handled or can be handled?
You understand? Look, what we're doing, we put some chemicals, this tranquilizer in the tank, okay? Whenever we see that the fish is quiet, or if we want to knock them down, namely totally sleep, we wait till this moment. They can be in this position when there is oxygen in the tank for long, long minutes. After you finish the process, yes, you move them to fresh water with oxygen, and plus minus the time which took them to go to sleep is the time which took them to wake up. But there are fish which are very sensitive to these chemicals. Silver carp, for instance, if you put dose too high, you might kill the fish. Small story about me myself. I have tried to select broodstock of common carp in tanks. So I put a lot of chemicals, this phenoxys and all the fish knocked down, went to sleep on the bottom of the tank. I chose a female which I wanted, and then put fresh water. Half an hour, no movement. I was sure I've killed the fish. I called from the market, the trader, the wholesaler, to come and get some very big fish, good for market. He came after an hour and a half, okay? We saw the fish start to move, and recovered from this tranquilization. So, don't take from me numbers. If you're using those chemicals, you have to try yourself in your conditions, okay? And if you, this lecture was recorded, there is a table which describes exactly which fish, the dose of which chemicals is suitable for tranquilizing. No need to put more than written in this table.
Okay, thanks. Okay, Buga, Asafo, your questions, let's leave them for next meeting, okay? So, but only something to clear up. Achik, you spoke before, and it was not clear if you spoke about pine tree or palm tree, because...
Okay, good. I was talking about substrate for spawning. Why am I saying kind of pine tree?
Pine
tree, because the leaves are very, very small. The area, total area of pine tree is very big, and the leaves are also last long time in the water. So in Africa, I know not every place they have pine trees, but any place they have roots of this lattice, what's the name? I don't remember. And you can find a natural substrate for spawning. I was in Kenya giving them training courses for catfish. And I started to describe, we take brushes of nylon and this imitate nature. And says, Achik, I want to show you what we are doing. They do the same spawning as I explained. They took these roots of the lattice, put inside, beautiful, and this is good enough. So everyone will find in its own place a suitable substrate, which has to be in a big area for lots of eggs and last long in the water. So I was talking about pine tree, yeah. Evelyn, you are on mute.
Sorry, okay, thank you. Jean-Michel says that he thought that pine tree was not good because of the resins.
No problem. Believe me, no problem. This resin is negligible. The eggs are attached strongly to the pine tree. No problem, no problem.
Great.
experience.
Great.
By the way, there are many other trees, I even don't know the English name for them, which are suitable for this purpose, okay? If somebody will have, would like to experience this thing, he will contact me personally and we shall find what is the best solution. By the way, in Israel, many people are using this bags of onion, which is made of kind of ropes, okay? Also good. Just big surface area for the eggs to be attached to.
Great, great. I think it was an exciting lecture. So I think we have to say thank you very much. Achik, bravo.
Thank you so much. And we should probably meet next week, same time, same hour. And I will try and get you the movie of spawning of ornamental fish, but it's not so important. So you see the stages one by one. Any question, we should discuss it next week. Okay?
I will send you some questions that are still here in the chat. And for sure, you are invited to stay with us. We are going to hear about Zambia.
I was in Zambia. I've enjoyed Zambia so much, you know, down in Kariba Lake, and there was a big company for cages, which I had to advise them. But unfortunately, fish are more important. I have to go to my farm now and make sure all the aerators are working before night in order not to get dead fish tomorrow morning. So have a great weekend, and see you in Zambia, Madagascar, and all over.
Great.
Next week. Bye-bye.
Take a little video of your fish so they say hello to us next week.
I will.
Thank you, Achik.
Okay.
Thank you
Bye-bye.
so much.
Thank you. Bye-bye.
Bye.
And now I'm leaving the meeting. I'm sorry. 1, 2, 3. Bye-bye.
Ciao, ciao. Okay. Good. You are fine. Everybody fine there. I want to see a smile. Reaction, a smile, a heart, a finger, something. Okay, good. Samantha smiling. Your smile always, it's good to see. Okay, friends. Thank you. We are going to Zambia. Yes. And I understand Chipepo and Mwangala will be with us, sharing about Zambia. Who begins?
Yes, please. Have you gotten the document?
Oh, no. I should have received them.
It's in the email. Carl sent it.
Carl, when did you send this? Oh God, no.
It's unfortunately a bit late, but today.
Yeah, it's just about 13:45.
Yeah.
By the way, can you hear me?
Yes. You can share it. You can share it, or shall I? Can you begin speaking maybe, and I bring it because I have to look it in another computer. I have it not here.
So I'm not familiar how to share a presentation on Zoom.
That's very easy. That's very easy. Bless you.
Let me try. Let me see if I can.
If you can try, that would be great.
You have to have it open before.
Yeah, I've opened it. So now I want to try sharing my screen, and then I'll
share screen, and you will see it when you open. And you say share, and it should work. That's okay. Great. Yes, we see it. Wonderful. Great. Excellent.
Okay.
Super.
Sure. So I'll give the project context, and my friend Mwangala will join in later. So our our project Fish for Food Security is actually under the agri-food cluster, agriculture food cluster that we have in GIZ. And in this cluster, there's the special initiative, the One World No Hunger, which is towards ending hunger and malnutrition, addressing the challenges that are in relation to that. So the projects that are in the agri-food cluster, most of them are also working in eradicating hunger and also ensuring that future generations have sufficient and affordable, healthy food in spite of the growth in population, the rapid growth. Yeah. So these are the 4 projects, as you can see on my screen here, the ones that are in the agri-food cluster. So there's the Green Innovation Centers, that's JIC, there's FANSA, there's AgFin, and there's Fish for Food Security. And when you look at the Zambian map here, you'll notice that 3 provinces have been highlighted. That's Luapula province, where Kao, Mwangala, and myself are. And there's Eastern province, and there's also Southern. Eastern province having all 4 projects currently running there, present. And we are also joined by AgFin, though they haven't yet sent a person up in Luapula, but we will have them here in Luapula. So we are working with FANSA in the agri-food cluster in Luapula, yeah, so far. And then just in short points, yeah, an overview. So the JIC, that as you can see here in the agri-food cluster, of course, the JIC have the small-scale farmers, processors, and agro enterprises as their target groups. And then AgFin, same thing, farms, rural agro enterprises. Then for ourselves, since we have in Eastern Province and Luapula, meaning it's different. In Eastern Province, they're actually working with dam committees, while in Luapula, we're working with aquaculture farmers. And so, yeah, artisanal fishers in Eastern Province. Then the FANSA's target groups, women of reproductive age and children below the age of 2. So you can see the common denominators there, small scale farmers, and also the fact that we're all working in food security, food and nutrition security. So there are these similarities. So it's easy for also collaborations because target groups are similar in most cases. Why fish? Well, fish is affordable, but not only that, it's very much widely consumed as a source of protein, of course. And well, in Luapula, you can speak for Luapula, it's the cheapest source of protein. The picture you're seeing here, in the most places, it's called Mbale or Chivale. So you get small fish on a plate. Normally, like right now, it should be going for 10 kwacha. So that's something that a lot of people can afford regardless of the income bracket. And at the end of the day, at least the whole family can actually afford to eat the fish. And well, as we can see, it's small fish, so mainly it's eaten whole, especially when they fry it. You can eat everything, so you're eating the bones, the eyes, everything. So you're getting the full benefits, and a family is able to feed just using 10 kwacha. That should be 50 cents, if I'm not mistaken, somewhere below 50 cents, maybe 40 something cents. Yeah. And also fish trade provides employment and yeah, a livelihood for millions of families. As we know, there's a number of stages in the fish value chain. If we're speaking of fish farmers, there's fish farmers, you still find traders, and the same traders also have levels of where they are found. There are people who are buying directly from the source or the producers in this case, and then there are people who now take that fish to the markets and sell it to. So some will sell it at wholesale prices, and then it will be distributed to the traders who sell it at, is it retail is also? Anyway, yeah, you get it. So you can see the lovely pictures. Oh, sorry, I think I moved this. Yeah, the ponds there. The objective of the project Fish for Food Security in Zambia is supporting the development of aquaculture sector here in Wapula, and then also in Eastern Province, supporting sustainable rehabilitation of dam-based fisheries. Yeah, and strengthening the dam committees that are responsible for the fisheries in those districts. It's a number of districts. The dams about 10. So yeah, that's quite a lot. While in Wapula, currently we are in Mansa, Kawambwa, and Mwense, but we intend to still extend to a few other districts, like Chipili, for example. So here, just a few points now on the projects. Excuse me. So the 4 main or key activities. Increasing aquaculture production is one of the main goals or the focus here in Wapula province. So training fish farmers in fish pond management, them adapting to feeding methods, and then also training them in business management. So that should help to increase sustainable aquaculture production in the region. And then when you speak of fisheries management in Eastern Province for the small water bodies, the dams there. So yeah, the local communities adjacent to dams as well as dam committee members, they get to benefit from the trainings of the sustainable fisheries management. And then also they effectively balance between the usage and conservation of fish stocks in those small water bodies and water resources. Well, training of trainers. Of course, just us as a team, we wouldn't reach the targeted numbers. We need people who can help us with that. So we have the multipliers, so such as the extension service providers that get to receive training, and they ensure the long-term availability of advisory services for sustainable aquaculture and also fisheries management. So we have had training sessions where we invited our colleagues from DOF, our partners, and also from ZCS, that's Zambia Correctional Services. These are farm managers in charge of the prisons, or the ones who also work with the inmates in farming, because most, if not all, the prisons, they have farms and some of them have ponds already. Yeah, so it's just a question of also helping the farm managers and the trainers to gain more knowledge in fish farming, because some of them actually don't have this. So with the help of DOF ourselves, we've invited them to trainings to help them with that. So yeah, and then promoting knowledge exchange, a multi-stakeholder platform is being supported to connect various stakeholders in the fisheries and aquaculture sector. So yeah, furthering cooperation and the implementation of practical approaches for the future development of this sector. So we intend to work with small feed providers, also some extension people within the province that are offering aquaculture services or other aquaculture training services, in whether it's supplying fingerlings or feed. Yeah. The outcomes that are, well, that we are hoping to achieve at the end of everything. Well, more fish, more work. So the production for aquaculture and sustainable fisheries management, well, it has increased already, and more people in the sector are provided with an income in their families. We have the fishers. Normally, you see, okay, fishers will be the men. You also find that the women are trading, so that increases income in households. And then sustainable fish. Well, when you look at the political framework in Zambia, it's improved to ensure the implementation of sustainable and resource-friendly aquaculture and fisheries. Then also, well, less fish from illegal and reported and unregulated fisheries. So like in the case of the dams in Eastern Province, having a case where there's more control and monitoring of fisheries in the dams, it has improved. And then, yeah, the capacities that are built to enable relevant authorities to come in. So all that helps. Then also fish for health and nutrition. So yeah, the nutrition situation of the target group, it also has to have improved. Like we are promoting not just eating fish, but the nutrition value to eat, the nutrition benefits. So we're not just saying, well, you should eat fish, but we're saying, okay, these are the reasons why fish is good. And in our trainings, we are also talking about the importance of the small fish, why it's good even for the children. We also speak about the first 1,000 days of the development of the child's life. So we promote the mothers eating fish. We talk about how the whole small fish will give calcium, iron, and that's good for the blood, the development of bones. And we also go in to talk about how they can use fish powder for the baby's porridge or include it in whatever it is that they are feeding the children, especially the ones under 5, which is the first 1,000 days. So yeah, all that. So this part now, Mwangala will come in. I think I'll continue sharing so that she can just.
Yeah, I think you should continue sharing. Okay, thank you.
Yeah, so you'll be telling me when to move.
All right. Okay, so right now we're looking at the small-scale fish farmers that we deal with in Luapula province. According to the World Fish Survey, we have about 1,000 to 1,500 number of fish farmers in Luapula. And most of these farmers have got very small fish ponds, which is fed by the spring, sometimes by water ground. And they have, most of them have shallow ponds with brick dikes, which is usually constructed by hands and helped by family members. And most of these farmers and their pond are affected by both the floods and the droughts. Yeah, most farmers stock up to 5 species, mostly tilapia, which freely breed in their ponds. And they usually yield very low fish for about maybe up to 60 kgs per pond. And this is mainly, at least, okay, it mainly contributes to a household protein security. And most of the reason why they yield this very small kgs from their ponds is because usually we don't have a lot of feed in Luapula, so they usually use just feed from farm waste, which is maize bran, vegetable leaves, sometimes just from manure from the animals that they are keeping. You can move to the second slide, Chipepo. Looking at the challenges that Luapula faces, one of the challenges is that it's a remote area and has got poor. It's poorly connected to urban centers. When very few farmers have been trained in aquaculture, then you find that most of the fish farmers when they start fish farming and they're not getting what they're expecting, they usually abandon their ponds. And most farmers have got a challenge with commercial inputs. It's very difficult to access feeds, quality pipes for pond constructions and the like. Even though Luapula has got these all these challenges, there are a lot of opportunities because we have like abundant water here in Luapula, and then we have abundant laborers, and the agricultural products are slowly increasing. And then in Luapula we have efficient eating culture, and a low livestock production. Markets are acceptably by small fish. Like Chipepo mentioned, most of the farmers will, most of the people in the community will be able to afford to buy what they call tumbalimbali as 10 kwacha, which will feed most of the family members. And as of now, commercial aquaculture is really growing in Luapula. So in the next slide, you can see here where in this other photo where you can see some people who are doing pond constructions, training with some fish farmers. This is a demo pond that we have at one of the institutes here in Mansa to just show how you can farmers can construct a newly improved pond. And here you can see some ponds which has poor nutrients, and then the other ponds are flooded pond dikes. You can move to the second slide. Okay, so in this picture, you see mainly when we are doing our trainings, we usually teach about the nutritional value of fish, and we have noticed that most of the fish, when we're doing trainings, most of the fish farmers here in Luapula don't really like the species spamini because it's a very small fish and it grows very fast, but does very well in fish ponds. Though most farmers think it will not give them much, they will not get much from it, despite it having high calcium and iron, and most people love it for it is very tasty. You can move to here on pond ecology. Most farmers in Luapula don't usually have fertilizer, manure, and most of them, the small scale fish farmers, can't really afford fertilizer to fertilize their pond. So we encourage them to use, they can even use grass straws, dry grass straws to put in their pond so that when it decomposes, it can form some microorganism which the fish usually feed on. And on the other side, you can see different diverse of species. You can see some algae, you can see some zooplankton in there, you can see some diatoms. And here we have a very green, well fertilized pond, which I'm sure which has a lot of algae from the process of photosynthesis and all that. Yeah, you can go to the next slide. Okay, so here you can see most of the farmers will usually assume that fish eats mud without them really knowing why without them knowing that the fish eat the mud because of the microorganism which is in the mud. So here you see a photo of a microscope and some farmers. Sorry for that. Here you can see some a photo with a microscope where we are showing the farmers the microorganisms that are found in the pond when it is well fertilized, and since we don't usually see these microorganisms by eyes with our naked eyes, you can only see it maybe with a microscope and stuff like that. Yeah, the next slide, please. Okay, any questions?
Okay, thank you so much.
Before Evelyn, just something to add actually.
Please, yes.
Yeah, so like when I was speaking about the same small fish and like us talking about the nutritional benefits of it, we have also noticed that a lot of fish farmers actually have an issue of having their ponds like overcrowded, and this is not a case of overstocking, but actually as a result of tilapia overbreeding. So since tilapia has a tendency to really breed, to like overbreed, we encourage them to crop out or rather thin out. So we encourage them to do this partial harvest. In this case, where they get to take out the small, medium sizes of fish that can be eaten, like you saw in the picture, so that they can leave their initial stock to continue growing with less competition over resources in the pond. So they get to benefit by not only remaining with more space in the pond, and that availability will help the other fish to grow, but also they get to sell a few the mbales, so they get to earn some cash, have some bit of cash flow in between, and then also they get to eat, so they benefit. They also benefit through the nutritional value of the small fish. So we are really talking about the partial harvesting a lot, because most farmers will just stock and forget. They think, okay, until maybe a year the fish should have grown, but because of the overbreeding, they find that the sizes really ain't changing. They have a lot of cases of stunting in the ponds. So yeah, questions now.
Thank you so much. So interesting. I don't know, maybe Carl, would you like to add something before we go to the questions?
Yeah, I think I can just maybe, Mwangala, if we could go back, Chipepo, go back one slide to the two pond ecology slides, the one before this. Sorry, thanks. Just
Which one do you want to cover?
Yeah, this one. Sorry, okay. Pond ecology.
This one, yeah.
Yeah. So, Mwangala, you've done a lot of the training with the farmers. Perhaps you could explain a little bit how farmers understand or misunderstand the reasons why to fertilize the water, and what they before they see the things with a microscope, what is their understanding for the reasons, and then again after seeing with the microscope. And then the other one was also that, with the picture of the stomach contents of the fish, again, that way you explain to the farmers around making that connection again back to the microorganisms, based on everybody having experience on cleaning the fish and seeing the mud.
Okay. All right. Thanks, Carl. So before the training, most of farmers think we encourage them to fertilize the pond because in that way the water is not clear for the predators to not see the fish. So they don't really get the concept of that fish actually, especially tilapia, actually feeds on the small microorganism that we don't usually see. And I didn't get your second question.
Goes with the next slide.
All right.
Again, with the idea that fish eat mud. Can you explain a bit on that, since that's often discussed in the trainings as well? Okay,
yeah. Most of the times when fishers or fish farmers or just the consumers, most of the times when they get the fish and they get to clean it, they see some mud, they think the fish eats the mud. So most farmers have got that perception of saying fish eats mud. But most of the times the microorganisms sometimes go on mud. You see some fish eating microorganisms, like eating mud, but they're not eating mud, they're eating the mud because of the microorganisms that's there. And sometimes you find that the fish will feed maybe along the grass, sometimes just besides the ponds. And also sometimes you see the fish maybe feeding by skimming just on the surface of the pond, especially when it's hot. So that's where the mud is coming from. Actually, the fish is not feeding on mud, but the microorganism that's on the mud.
Interesting very much. It's okay. Let's go to some comments. Carl, it's fine with you?
Yes, thank you. Or maybe one more, the top picture on the top right, maybe Chipepo, can you just explain to the participants what's happening with that chart there and why we use it?
Okay.
We can move on to
Sure.
questions.
All right, sure. Sorry, just the top, just the charts. Anyway, okay. So just for context, anyway, I'll actually try to give a small highlight on the pictures. So this picture here, this is in Mabumba. We were having a pond-side training. Those are usually fun because we get to, like Mabumba, where the ponds are in one area, but they all belong to different farmers. And even though they're in the same area, sometimes each farmer will have their own way of managing their pond. So it really helps if we're doing some comparisons, describing, okay, here the water seems this way. You see, maybe this pond has a lot of fish and all that. Then here we have the fish chart. So this fish chart, we actually give it to our farmers after trainings, the trainings that we were having before the COVID issues. We would usually have like a group of 10 farmers, but also we take to the ones that we meet in the field. So we give them this chart, and it's a good resource for sampling. So as you can see there, you have the ruler at the bottom, so it's actually very accurate. And then this, you see the, it's like the fish keeps extending. There are like so many sizes of fish, and at the tail there's a number in grams. So and it's laminated, so you can use it with water. So we use it for sampling to tell the grams. So a fish that's just growing at the right growth rate will actually, we've found that it will be able to tell us if it's a 10-gram fish, 20-gram, whatever, it's 300. We actually have a small scale that we also move it. So we make sure, okay, when we do use the chart, we'll maybe even weigh to just show the farmers how accurate it is. And the farmers have really, really loved it. The ones who have not gotten the chance to actually meet with us have been requesting for it. So this has been very helpful, not only with us, but also the farmers. And we know that it's an issue for them to buy scales. Like most of them, the level of fish farming, they feel like buying a scale would just be too much money to be spent on that. So these have come in handy. Yeah. And then here when we had our operational planning meeting in February, it should be. So here we were with Dove, and we were having a training with them on just throwing cast nets, using cast nets. We actually had an exercise after that. Luckily, while we had the operational planning meeting, they have ponds, so we all got to try our luck at getting some fish. Yeah, and that was fun. Then here, well, just another one of our trainings where we'll use the projector and show them everything happening in the pond water using the microscope. Thanks.
Great. There are a lot of, there are some people saying, I'm sure actually that many people are interested in that chart. Is it possible to share it in the WhatsApp group or the picture? So everybody.
I think the picture won't be accurate unless they just want to look at it. But if they want to use it, they would have to get the physical one.
It's well, we can share it by email also. It's important that it has to be printed to the exact scale. Otherwise, otherwise the weight length relationship won't work out. It's based on some averages between the different species so that it's relevant for whatever fish we find among the tilapias in the ponds. So of course there might be some small differences if the condition of the fish is different also, but for farmers wanting to understand their sizes, do their sampling, it seems to be working quite well.
Yeah.
Okay, so if it's possible, it will be nice to share it with everybody.
Yeah,
Yeah, thank you, Chipepo. Maybe somebody else from Zambia would like to share any idea, any comment, any experience from the group. In general, we have some people managing farms. We have people from the university, from Nosobe fish farm. Who would like to share with us any comments from Zambia? If you can stop sharing.
Hello.
All right. Wilbrot, please.
have I stopped sharing?
Thank you. Thank you.
Yes, good afternoon. It's a good place. Hello.
Yes, let's see if we can hear you. Yes.
Okay, yeah, yes. Good representation. I'm running a fish farm and I've taken that concept.
Mr. Msanji.
Wait, Wilbrot, we don't hear you well again. Can you try?
And we can see the result just be a lot.
Yeah, we cannot hear you well. No, no, sorry. Please write it on the chat, okay? Great presentations. There are very nice comments. Well, there is a question here. Let's ask for everybody. Hopeful. Oh, we have hopeful. Hopeful. Would you like to comment directly? Ask.
I think the question has been already addressed by Carl, because I was asking about if the farmers from Luapula are able to sell the fish to urban markets, but he has said that most they do sell to their neighbors and just a few into towns. Actually, I was looking at how the farmers can make a lot of profits, because if they're going into commercial fish farming, then they need to know their markets, something like that. Thank you.
Okay, good. Well, Colin, please.
Thank you. Good afternoon, and good afternoon all. Hello.
We hear you.
Yes, you're
okay. Okay, thank you. First of all, I would love to appreciate the presentation that has been done by GIZ Luapula. That is wonderful, and I can say myself, I'm actually a beneficiary of that scale that many people are questioning. I actually run a company, and we grow out fish. That is Unimos. Now, that scale, when the first moment that I got it, I began using it, and it has given me a concept on which I should only withdraw fish when it has attained 300 grams. And this has enabled me to understand how many fish or how much I need in terms of the kilograms per pond. I can plan before I harvest. Let's say I have got ponds, each one worth 4,000 fingerlings. So I know that if these 4,000 fingerlings, they are grown up to 6 months, they are able to give me 1.2, that is 1,200 kg. So all because it is because I use that scale. I don't normally take, like whenever I'm taking samples, I don't use the scale, the digital scale to put there. But if I use just that scale, which is on paper, I just measure, I'm able to get the 300 grams of each fish, even above, within a period of 6 months. So I'm attesting today that that scale, it is very helpful, and we are using it. Concerning the market in Luapula province, Zambia, the way it is located, Luapula province was known to be the best producers of fish because of the lakes that we have in Luapula. Now, when we look at the lakes today, they have no fish, and Luapula supplies to the Copper Belt and Lusaka province and other provinces, I can say. But of late, we see that fish has been depleted from the major lakes. Now, whenever that we harvest the fish, in most cases, you'll find that the fish will finish just from the farm itself without even taking them to the market. But even if you take again to the market, the fish that you produce is more tasty than the fish that is being brought or imported into the province. So this fish that we produce has more market than the other. So you find that it may not take a day before you finish selling the fish. So all we need to do is to upgrade our level of production so that we may be able to supply to supply the demand which is there. Thank you.
Thank you very much, Collins. Veron, you raised your hand before, but I don't know. Are you?
Oh, yes, hello.
Yes, hello. Please.
Yes, I'm Veron. Yeah, I'm also a beneficiary of the same trainings under GIZ Luapula. I think, as you saw from the presentation which Chipepo was making, or she was making, I think one of the issues she brought out on the challenges was on dam construction. I think from the training, I've acquired enough skill. At least we are able to construct a nice pond. I think I have some pictures which I would like to show. Let's hope, I think I will share later on, but on how we are doing our ponds now. Thank you.
Okay, thanks. Oh, good. We have a chicken house there. Mr. Collins, you have a question from Asafu. Is Eunimus or any other fish farmer doing any fish value addition? Collins, did you get me?
You may repeat the question, please.
If Eunimus, you're from Eunimus, no? Or
Eunimus.
Eunimus, or any other fish farmers, yeah, sorry. Are you doing any fish value addition? And Carl is answering, but you are invited to answer.
Currently, we have not yet started, but our program are that we begin doing the fish fillets and also canning the fish. So that is our plan in terms of value addition to the fish that we produce.
Mr. Chongo, you do freeze or cool the fish before selling, right?
Yes.
So that's already the first step of value addition. If it helps
Thank you for
get a better price or helps the fish to sell faster, then that's the first step. But good to hear. You said canning, and what was the other one?
Making fish fillets.
Fish fillets. Okay. Ooh. Yeah, great.
Yeah. Then at the same point there where you mentioned freezing, we have opened an outlet which is in Chembe district. So there you find fish that is direct from the ponds. So I think we are not mainly running out of fish in our outlet in Chembe. So many people are accessing our fish products in Chembe district.
Thank you, Collins. Mary, you have your hand raised.
Yes, thank you for the presentation. And I just wanted to add that we have another farmer who is doing value addition, although he's not in this meeting, to the Zambians, Triple Blessings in Luwingu. So he is buying fish from the farmers and he freezes them. Thank you.
Thank you, Mary. And looking here at the list, I see we have some people dealing with training and education, people from the university, from Kasaka Fisheries Training Institute, Mansa Trades Training Institute, colleges, natural resources colleges. Maybe you can share with us a bit about your challenges. What happens with the young people? Are they interested in fisheries, in aquaculture? Is it easier to introduce innovations with them? Who can share a bit about that? Do we have somebody here with us?
Yeah, I'm Musuka from Kapasamakasa University in Chinsali. Yep. Yeah, we've been training students the last 12 years who are doing undergraduate studies. That is a Bachelor of Science in Fisheries and Aquaculture. And since we started, our enrollments have been very, very overwhelming. At the same time, those that have completed, some have joined the government, but some have gone it alone. They've become entrepreneurs. I have one example at just our next university, Mulongoshi University, just opposite Mulongoshi University. We've got one young man who has ventured into fish seed production. His company is called the Mukasa Agro Solutions. So he produces catfish, he produces tilapia, which he sells to other people across the country. He's been quite an enterprising young man. And also as a university, we do conduct some short courses for farmers around as part of our social community social responsibility. The last 3 years, we have trained over 300 fish farmers in various, in the basic fish farming activities. At the same time, we've made use of some of our former students to come and we incorporate them in our training activities. Besides that, we have others again who have gone into other areas, but within the aquaculture sector, we have 2, 3 who are in feed production. One of the big producers of feed, which is known as Novatech, they have a section which deals with fish farming as well as the fish feed production. So he's also involved in research. Besides research, he was also conducting some training activities. And then we have others again who have gone into other value addition, like other people have mentioned. They've gone into other activities. In Luapula, we have one of our former students who is running a hatchery, a fish hatchery. So he's producing seed for the farmers within the catchment of Luapula. So there is quite a lot. There's a lot of enthusiasm amongst our students, current students, as well as those who have graduated, who have not found jobs, but they've opted to go it alone and go and become entrepreneurs. So we are happy with that. And even when it comes to enrollment, we've never been short of students who have come for our programs. In fact, just to add on that, we're trying to introduce another program because we've just been running a BSc in aquaculture, fisheries and aquaculture. But coming next year, we are introducing two new programs, one a Bachelor of Science in aquaculture, as well as a Bachelor of Science in fishery science. So all this is coming as a result of the overwhelming response that we are having from the would-be fish farmers or fish entrepreneurs or government workers or policy makers, as it were. Thanks so much.
Good, great, great. So I understand you are working all together very good, and I hope this little webinar will help you a bit more to join efforts and work together. Yeah, Mary, you want to add something?
Yes, I just wanted to add something on the youth. Of course, before it was very challenging when, you know, they could hear about fisheries or aquaculture. They didn't know like exactly what was in there. So for the past years, it's been very difficult, as Musuka has pointed out. But now I think people are realizing that once, for example, you do aquaculture and fisheries, you are not going to catch fish like on the lake or from the river. There is something you can do. And so in line with that, World Fish as an organization, we've also helped in that line by upgrading the curriculum at one of the colleges so that, you know, they get to have the up-to-date skills. So we've put up a hatchery at that college and a field training. There's a hatchery, ponds, and also there's a computer laboratory. And we provided computers for online courses, long-term, short-term, and this is attracting the younger generation to major into fisheries and aquaculture. And so the graduates from that college, some of them are working with feed companies or hatcheries, private hatcheries, and some of them we've employed as interns or on permanent basis. Those are the young people, both girls and boys. And so I think it's opening up. They are seeing the importance of that. Even looking at what other youths, for example, Collins Chongo there's another one, Mukasa, who Musukwa talked about. So they are seeing how they are doing and they are motivating them. So I think they are coming on board. Thank you.
Great, great. Thank you very much, Mary. One other thing, you know, here in Israel, we always speak about the holy triangle. We are speaking about the joint venture, let's say, of the research, the extension system, and the farmers. So we have somebody from the research here with us, which is Kondwani Goma. Kondwani, would you like a bit to share with us what you are doing in your center? Some challenges, maybe problems or success? Let's see if you have.
Hello.
Yes.
Yes. You can get me?
Yes, we hear you.
Yes, we hear you. Well, as the agriculture station, our objective primarily has been to supply the province. The challenge that we've had is farmers do not have a place where they can get quality seed. So amongst them, the objectives that we have is to give them fish seed. And amongst the things that we do we supply Coptodon and Orochromys macrocha.
I think we hear you. I don't hear you very well. I suppose we don't hear you, everybody. I don't know. Oh, so maybe you share with us something in the chat. Okay, it's
okay. It's very. Yeah. Can you hear me now?
Let's try. Let's try again.
Hello.
Let's try.
Okay. So I was saying, amongst the services that we offer as a station, that we offer fish species such as Tilapia rendeli or Coptodon rendeli and Orochromys macrocha. We've also been trying to breed the giraffe catfish, which is Oceanoglanis occidentalis, of which we haven't had much literature or study on it on reproductive biology. But currently, as what we do is we've been having an overwhelming response in that people have been requesting for fish seeders. To be precise, been requesting for the challenge to actually meet the demand in the province. I think for now, that's all I can, unless there are more questions.
Okay, great. I think, please, again, our WhatsApp group, I know our friends from Madagascar are not there, but we could copy actually the chat to the email and send you. So please, again, I think this is a great framework to share comments, experiences, news, okay? So please feel free to do it. Friends, we are at 6:15. What do you say? Shall we split into groups, or it's okay for today? Somebody can help me with this?
It's okay for today.
It's okay. No, I also think so. Okay, so I
agree.
I would suggest, again, next Thursday we meet again with Achik, and the subjects are fish reproduction and fingerlings. So please, if you have questions, let me know so that I can send the questions to Achik, and we will have 10 minutes, 15 minutes maybe at the beginning to answer them, okay? And if there are some other ideas from each country, please share them. Fine. So, friends, it was great. Somebody wants still say something, share something, maybe Samantha or Carl or somebody. Would you like to comment something? No?
I can, or Samantha, are you there? Would you like to?
I'm here, but I was shaking my head, so no, go ahead, Carl.
Okay, no, great. Just thanking everybody for your interest in our presentation and in joining us in taking a look at the context that we are working in. There were some interesting questions, and I also hope that the background in fish reproduction that was presented today, even though it was quite technically detailed, There might be topics that you are very interested in. Please use the network to give us feedback if there's something you've seen that's very relevant for your interest, whether it's a farm or whether you're an extensionist, or with regards to fish reproduction, any topics you want to understand better, because from what you've seen today, please get in touch with the different leaders in your countries on this program, and we can take up that discussion. Thank you very much.
Thank you, Carl. Maybe we have somebody from Malawi who would like to add something. Mara, you are with us, Mara?
Yes, I'm here. Asafo, since you are our country representative, I'm sure you also want to say 1 or 2 sentences. I think you already posted in a chat that we will collect some questions and comments again in our Malawi chat and then forward it to you.
Yeah, thank you, Mara, and thank you, Everine, and the team. Thank you for the wonderful presentations which we have had today from our colleague from Israel and our brothers and sisters from Zambia. For the next session which we are going to have, I think as a team here in Malawi, we'll compile questions and comments and we'll share them to you so that possibly they should be answered in the next session which we are having. Because here in Malawi, we are dealing mainly with the tilapias and catfish. So we'll sit down, brainstorm, and share questions and comments, which will be posted to you within the week. Thank you.
Great, great. Thank you so much. And maybe somebody from Madagascar would like to say something. Jean-Michel, I think you are not with us at the moment. Somebody would like to say in French, maybe? No? Nobody from Madagascar? I understand you are a bit in problem for the moment.
Oui, en fait, je vais parler en français, si vous voulez bien.
Ok, je ne parle français un peu, but mainly for your group, Phiri, maybe, please.
Je crois que
Vous
c'est bien.
pouvez parler en français, oui.
Oui, oui, merci.
Ah, Lara est here?
Merci.
No,
Merci beaucoup. Merci beaucoup pour toutes les interventions d'aujourd'hui. Ça nous a permis de savoir un peu plus, étoffer nos connaissances sur la reproduction des différentes espèces de poissons et aussi les expériences des autres pays sur la pisciculture, les pratiques des pisciculteurs, que nous allons aussi voir la possibilité. Par exemple, on a été vraiment étonnés sur l'utilisation du pain, l'arbre pain pour le support de pente, mais on va expérimenter de notre côté si c'est possible et faisable parce que les pins existent beaucoup chez nous. Donc, ça peut être une solution durable pour les pisciculteurs et accessible. Et donc, c'est un exemple parmi tant d'autres, mais nous vous remercions beaucoup de ces interventions et on espère la prochaine fois avoir encore un peu plus d'échanges et aussi avec Madagascar. Merci beaucoup.
Merci, merci. I will ask Clara then to translate, okay? Don't worry. Samantha, would you like to say something?
Oui, seulement pour nos collègues en Madagascar, c'est pas un problème aussi de poser des questions vers l'équipe en Zambie, parce que nous sommes au moins 2 personnes qui parlent français.
C'est très bien. Ok, c'est bon à savoir. Merci beaucoup. On ne va pas hésiter.
Merci.
Great, great. And for sure, I hope you will learn a bit of Hebrew. Maybe next time we will share some words, okay? Did I tell you how we say fish in Hebrew? You say duck.
No.
Duck. Here in the chat, duck.
Duck.
One fish, duck, and many fishes, dagim. Dagim, okay? So, dear friends, dagim, it was great to see you today again. I think we always learn new things, and that's the important thing. So thank you for being with us, and have a great weekend, and see you next week. And we will be in touch for sure.
Thank you.
Thank you.
Thank you.